Okay, so we were discussing about the DNA replication. I said that you should know all the definitions here, right? So for example, when we say origin of replication, what is origin of replication? It's that part in the DNA where the replication will begin, where the replication will start, okay? Usually very rich in adenine and thymine as compared to guanine and cytosine, okay? And then what is the replication fork? Okay, so replication fork, as you can see here, once the DNA is opened up, right, you can see the replication fork is formed. It is the place where exactly the replication is taking place right now, right? Yes. Okay, so this is a replication fork, as you can see over here, okay, very clearly. Okay, then you need to know all the important points of the DNA replication, right? You know, we said that replication is semi-conservative, point number one, right? I showed you that, right? One old strand and one new strand will always be there. One parent strand and then one new strand will be there, right? Replication is bidirectional. That means it can take place on this side as well as this side. It's usually bidirectional in the eukaryotes, okay? And then I said that replication begins at the origin, right? This is the ori site. This is called as an ori site, okay? So then you have to know that number two, you have to know that about the leading strand and the lagging strand. Okay, so what is the leading and the lagging strand? If you remember, we discussed it previously that I told you you need to remember the read up and write down formula, right? So DNA polymerase, which is the main enzyme, remember, forming the DNA, right? Which is involved with the replication of the DNA. It reads up the DNA strand always in 3 to 5 dash direction, but writes down in 5 to 3. So DNA strand is always formed in the 5 dash direction to the 3 dash direction. So then what is the leading and the lagging strand? Leading strand is that strand of direction where the DNA is synthesized continuously, as you can see here. Here the DNA is formed continuously. Okay. So this strand is called as the leading strand. This newly formed strand is called as the leading strand. Okay, so which is continuously polymerized or formed. While as on the other hand, as I told you that DNA polymerase reads only in 3 to 5 dash direction. So DNA polymerase is going to read this strand, it's going to bind to this strand, right? It's going to read it in 3 to 5 dash direction. But writing it down, that means forming the DNA in 5 to 3 dash direction, right? So that's why on this strand, the synthesis is discontinuous. Okay, so it is called as a lagging strand. So this strand is called as a lagging strand, where you have smaller fragments of the DNA being formed, which are called as an Okazaki fragments. Okay, yeah. And then one more point you have to remember, that DNA replication will never ever take place in absence of primer. We need a primer. Do you know what is a primer? We discussed that, right? Before we form the DNA strand, we need a primer. If you remember, I have a diagram also for that. So this is a table. You memorize this table for all the enzymes. Okay. So what is an RNA primer? If you remember, I will show you here. There's a diagram. Look at this. We need a primer to be formed, right? So what is the primer? Primer is simply, which is a small oligonucleotide sequence. What is an oligonucleotide? That means, very few nucleotides you have there, like 4 to 15 nucleotides, right? And very important point to remember, primer is always, made, it's an RNA primer, not the DNA primer. So primer is always an RNA primer, not the DNA primer. Okay, this is a very important point to notice, right? So then what happens? Then we have an enzyme here called as a primase enzyme, DNA primase enzyme. Yes, synthesized by the primase. You're right, Mohammed, right? So primase is the enzyme, okay, which actually synthesizes the what? These primers, okay? And these primers are required for the DNA synthesis, because if this is a primer, let's say this is a primer. So this is a 5 dash 3 dash end. So it's going to provide us this 3' end. So the new so the new nucleotides will be added over here like this, like this, like this. So extension of the primer would take place, right? So that's why we need a primer, because DNA polymerase will not be able to synthesize the DNA strand unless and until primer is present. So primers are very, very important. Okay, and later on these primers are removed, as you can see here. Once we are done with the DNA synthesis, primers are removed, okay, by the exonuclease activity. Mean, the cutting activity, the way we have a scissor to cut something. So DNA polymerase also has an exonuclease activity. It removes these primers, and the DNA strands will be joined back together by the DNA ligase. They will be first filled up with the nucleotides, and then they will be, wherever there's a cut, it will be joined back by the enzyme called as the DNA ligase, right? So with that, we just discussed two enzymes, one the primase enzyme and the DNA ligase enzyme. Primase is the enzyme which forms the RNA primers. Ligase is the enzyme which joins the Okazaki fragments or joins any cut in the DNA or in any nick in the DNA. Then what is the function of the helicase? If you remember that the first and foremost step, okay, in the DNA synthesis or in the replication is you need to open the superstructure of chromosome. We know that we discussed about, the histone proteins, okay, and the DNA is wrapped around those histone proteins, right? So we just discussed about that in the previous class. So people who have joined us just now, they can go back to the previous, video which I will be uploading on the Moodle. So it's a revision class. Okay. So how do we open the structure of the DNA? The supercoiled structure of the DNA. How do we open it? How do we open the super condensed structure of the DNA so that enzymes can easily access it? How do we open it? Do you remember, anybody? Acetylation and deacetylation, remember? Okay, so simply there's an enzyme called as the histone acetylase, which causes the acetylation of the lysine in the histone proteins. Do you remember I told you that histone proteins have the special amino acids like our lysine, okay, which have the positive side chains. Okay, so it causes acetylation of the lysine present in the histone proteins. Okay, so once the acetylation of the histone proteins is caused, it removes the positive charge of the histone, isn't it? If you remember, I told you that histone proteins, right, they are all what? Positive in charge. Positive, positive, positive. And DNA is all negative in charge. So unlike charges attract each other, that's why they are together. But right now we need to open them because we want the enzymes to do the replication, to access this DNA. We don't want histone and DNA to be together, right? So we need to remove this positive charge of the histones. And the only way to remove the positive charge of histone proteins would be causing acetylation of the histone proteins, which will further decrease the activity between the histone proteins and the DNA. So this interaction will be decreased. Is it clear? And remember, the enzyme is the histone acetylated transferase enzyme, or just the histone acetylase enzyme. Okay, so it basically simply weakens the strength of the DNA histone interaction. This is the first step. Okay, and then the second step, if you remember, I showed you a video also last time. Okay, when we are removing or breaking the hydrogen bonds, unwinding the DNA, right? So helicases are the enzymes which are called as the DNA unwinding proteins, because they break these hydrogen bonds present between the bases. Okay, they break the bond between adenine and thymine, or guanine and cytosine. Okay, and as a result, the DNA strand will be separated from each other. It will unzip the DNA strand, which will form this is a replication bubble formed. So as it's unzipped, it will form the replication bubble. Okay, so what is the function of DNA helicase? Just to unwind the DNA, break the hydrogen bonds. Okay, they attach themselves to the DNA strand. As you can see, helicases are here. But then once it is breaking these bonds, there is a possibility that we can have these supercoils, the positive supercoils formed. Okay, then comes another enzyme called the topoisomerase, also called as a gyrase. We have two types of topoisomerase 1 and topoisomerase 2. One can break only one strand of the DNA, while a topoisomerase 2 can break both the strands of DNA, can remove these supercoils. Okay, are we okay? Did you all understand function the topoisomerases to remove this supercoiling and relaxing the DNA? So this was the topoisomerase, this was the helicase. Okay, and then we have the clamp protein. Okay, clamp protein, if you remember, I showed you in the last time in the diagram. Look at this. This is a clamp protein. Its function is to hold the structure of the DNA, okay, while the nucleotides are being added. So holding, okay, attaching, or you can say, bracing it to the DNA strand is the function of the clamp protein. Okay, and then the very important enzyme of the replication is basically the which enzyme? It's the DNA polymerase. Okay, then we discussed about it needs what? It needs a primer. We discussed about that, right? So what is the function of DNA polymerase? DNA polymerase simply synthesizes the complementary strand of the DNA, right, in 5-3- direction, okay, by using what? The template DNA. Look at it, uses a template, okay. Is it clear? So, so the DNA polymerase has a capability of, incorporating the bases which are opposite to each other. Adenine will have thymine, guanine will have cytosine. Okay, so if we have adenine present in the in the template strand, here we'll have the thymine in the new strand. This is all done by the DNA polymerase, right? So it helps in the synthesizing the DNA synthesis. Okay, and at the end it forms the phosphodiester bonds between the nucleotides. If I have one nucleotide here, another nucleotide here, another nucleotide here, so these phosphodiester bonds are also formed by the DNA polymerase enzyme. It not only adds the nucleotides to the 3-OH end of the primer. Look at this. This is the primer where the new nucleotides are being added at the 3-OH end. But it also helps in joining those nucleotides together, forming the phosphodiester bond between them. Okay. So, and then we already discussed about the leading and the lagging strand. We already discussed about that. And the ligation, what is the ligation? As I said, that finally joining the Okazaki fragments or joining any cut in the DNA is the job of the DNA ligase. So the rules of the replication, it's a semi-conservative, starts at origin, can be it's usually bidirectional in the eukaryotes. Semi-discontinuous, right? The lagging and the leading strand, and always synthesis takes place in 5-3- direction. Remember these rules, and RNA primers are required. Okay, and then that was about it. Do we have any question about the DNA replication to everyone? Are we okay about it? Any questions about it? Then we can start discussing the PCR. Yes, ladies, all okay? Boys, ladies. Okay, great. So then PCR, you should know what PCR is, right? The definitions, you should know the steps of the PCR, the requirements of the PCR, right? So if you remember, I told you that PCR was a very important, diagnostic, basically instrument created by Carey Mullis. Okay, he was the one who created this thing. Okay, and the main purpose of the PCR is simply to amplify the sample to make more and more copies of the DNA, right? So you can say that PCR is simply the replication, but where? In the instrument called as the PCR, right? In this instrument, this. Replication is basically the in vivo technique wherein the replication is present within the DNA, within the cell. But here we are doing replication in the PCR machine, right? Which is used to amplify the DNA. Okay. And then what are the requirements of the PCR? What do we need for the PCR? We simply need a PCR machine, of course, right? We need the primer, of course. We cannot have replication without primers. We need the target DNA. Okay. What else? We need a special enzyme, the Dna Polymerase. If you remember, I told you that the specialty about this enzyme is a thermostable enzyme. If we increase the temperature in the PCR, it's going to stay stable there. That is very, very important, right? Because the bacteria from which we isolate this Dna polymerase, that bacteria stays at hot springs at a very high temperature. Okay. And then of course we need some buffer and all. So those are the requirements. So you should know what is the PCR, what is the purpose of the PCR, okay, what are the requirements of the PCR, and now the steps of the PCR, right? So in the PCR steps, what did we say? That we said that it has 3 important steps: denaturing DNA, annealing DNA, and extending DNA. Denaturation, annealing of the primer, and extension. In denaturation, we simply increase the temperature to such an extent, 95 or above, for 3 seconds or more, okay, wherein the 2 strands are separated from each other, as you can see over here. Step 2, we decrease the temperature to maybe 70 degrees, okay, which causes annealing. That means binding of primer to the DNA strands. As you can see here, one primer I have it here, and another primer I have it here. Okay, so second step is annealing of primers. Temperature is lowered. So remember the temperatures as well. Remember the steps, remember the temperatures as well. Okay, so in this step, the primers bind to their complementary sequences. And what happens in the third step? In the third step, the DNA is being extended, the extension. We add the nucleotides. Yes, the machine adds the nucleotides. Okay, the temperature is basically, between 60 to 70, 65 to 75 usually. Okay, so stick to the temperatures present in your textbook. Okay, so nucleotides are added in at the 3-OH end of these primers, and primers are extended. That's how from 2 strands I now have what? 4 strands of DNA. From 1 molecule of DNA I have 2 molecules. From 2 I have now what? 4. From 4 I might have 8. Similarly, it's going to be more and more and more till billions and millions of copies of the DNA are formed. So that is about the PCR. So you can read all these things, okay? And then the DNA repair, right? So why do we need to repair the DNA? If you remember, I told you that there is the possibility that DNA polymerase, when it synthesizes the complementary DNA, right, it can make mistakes. Like everybody makes mistakes, right? We all know that we do make mistakes, right? But DNA polymerase has a proofreading activity. What is proofreading, right? Once the new DNA strand, for example, if this is the DNA, old DNA, and once another color, once the new strand is formed, right? So what is proofreading? Proofreading is like, checking what did I make, okay? So that the process called the proofreading, right? So if the DNA polymerase detects that, oh, there is an incorrect base pair, I had to, for guanine and cytosine, I had put guanine and adenine, which is an incorrect base pair. So it will not only remove, it will but also replace it with the new base or a new nucleotide. Okay. So many errors made by the DNA polymerase are corrected during this proofreading activity, which is done immediately after the DNA replication. Okay, but sometimes you tend to make mistakes, right? There are some errors, which are not identified by the DNA polymerase, or which slips through. So at that time, what will happen? What will happen at that time? So then we have the DNA repair enzyme. You still need not to worry, right? we still have the DNA repair mechanisms in our body which can correct the mistakes made by the DNA polymerase in the new strand. Okay, but if this mistake is not repaired by, you know, as early as possible, right, then it can cause various mutations in the cell, right? And the two basic types of the repair mechanisms, the base excision repair and the nucleotide excision repair. What is the basically what is the difference between the two? In the base excision, if there's a wrong base which is incorporated, okay, as you remember, I told you that this diagram is very easy to memorize, right? If you see here, this whole thing is a DNA strand, a polynucleotide strand, and this is uracil. Oh my God, your DNA polymerase did a big mistake. Uracil was incorporated here. We don't have uracil in the DNA, right? So then we have an enzyme called as the G-glycosylase. So it will scan this whole newly formed DNA strand and it will look for the damage. And once the damage is identified, recognized, as you can see here, this is removed. Uracil is removed from the newly formed DNA, right? So as it's removed, so we have an empty space over here. As you can see here, there is no base, no base here. So we have phosphate, base, sugar, phosphate, sugar. There's no base here. So this site, an empty site, is called as an AP site. No, primer is not at this stage. Primer has already been removed. Remember the exonuclease activity, Mohammed? I told you when the new DNA strand is formed, then the DNA polymerase itself has an exonuclease activity, and that exonuclease activity of the DNA polymerase removes the primers. Do you remember, Mohammed? I said that when I was explaining, how primers are being removed. So that's not being done over here. Okay, that's not being done over here. That's done before you proofread, before you proofread, that's done right after you finish, extending the primers, and then we need to remove the primers. Then only the replication is complete. So this comes under the DNA repair mechanism. Okay. So when you remove the incorrect base, so there is an empty site which is produced, and that empty site is called as an AP site. Very important. What is an AP site? A purinic or a pyrimidic site. That means there is no purine here, no pyrimidine here, no base over here. Okay. Then as you see here, then comes another enzyme called an endonuclease or an exonuclease enzyme, which cuts the backbone, the sugar phosphate backbone. So if you remember, I told last, endonuclease cuts it in the middle and exo at the end. Okay. So once this whole nucleotide is removed, what's going to happen then after that? Then DNA polymerase inserts the correct nucleotide over here. Okay, whatever is required over here. And ligase will seal the gap there. That's how it was repaired, or it was fixed, right? So in the base excision, just a damaged base is removed. And in the nucleotide excision, you remove the nucleotide. You might remove 20 nucleotides, 30 nucleotides, right? Depends upon the length of the DNA. That is the difference between, the base excision and the nucleotide excision. Here's this one. So base excision, you just remove the base, and nucleotide excision, you remove almost 20 to 30 nucleotides. Okay. So that's what it is. And then we discussed about this disease. Okay, what is this disease called as? Xeroderma pigmentosa, right? We said that this is basically a genetic defect, okay, wherein the nucleotide excision repair pathway is mutated. The enzymes which can repair the DNA is mutated. So as a result, these patients are very sensitive to the UV light. So as a result, they have the pigmentation, okay, or tumors or skin cancers. Okay, they get severe sunburns, okay, because usually whenever we are exposed to the sun rays, so because of the UV rays, there are a lot of, damages which are being done in our DNA. But alhamdulillah, as long as we have these excision repair mechanisms in place, we don't need to worry about it. Okay. But for these people, the problem is this pathway doesn't work. So that is why the mutations keep on accumulating, 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 right? When they're exposed to the sun rays, right? That was about, this is all done previously when we had a class. So that's all about the first chapter called as the nucleotides. Do we have any questions about it, ladies and boys? Then I can start revising another chapter, the gene expressions. All clear? Okay, alhamdulillah. So I'm going to pause the recording and then let me upload the gene expressions chapter. Now let's start revising another chapter, gene expressions and the protein synthesis. Again, this chapter is for the midterm 3 as well as for the final exams. Okay. So in this chapter, if you remember, we spoke about mostly about transcription and translation. Like this is the main theme of this chapter, discussing about transcription and the translation. And then at the end, we discussed about the recombinant DNA technology. Okay, so we know the transcription is when DNA forms RNA, it's the transcription which takes place in the nucleus, right? And then mRNA goes out and it is translated into the proteins, which takes place in the cytoplasm, right? The central dogma of the molecular biology. And then we just said that we should know the difference between the replication and the transcription. Okay, how DNA was synthesized in the previous chapter, which we discussed a while ago, and now how the RNA is synthesis. Do we have any differences between the two? Very important, you should be knowing about this. Okay, so in DNA synthesis, you know that we were in need of a primer. So DNA, the replication will not take place if there is no primer, right? But primer is not required here in the mRNA synthesis. No, in RNA synthesis, we don't need primer because the enzyme which we are going to use here, the RNA polymerase, it can work without using a primer, right? It's only the DNA polymerase in replication which needs a primer. So here in RNA, we don't have thymidine, we have uridine, right? And there we basically we used both the strands as a template and formed the new DNAs, okay, in the complementary DNAs in the in the replication. But here you will see only one strand of DNA is used as a template for the RNA synthesis. There we saw that the entire GNA should be replicated, but here only one portion of the genome is copied, right? Only one part of the DNA is copied. And there we had the deoxynucleotides used. Here we have the ribonucleotides which are being used over here. Okay. So transcription results in the formation of a single-stranded RNA, but there we formed a excuse me, double-stranded DNA. Is it clear to everyone? You should know the difference between the replication and the transcription. Read the table. Okay. Then we already defined what is the transcription. And the types of the enzyme, the main enzyme which is responsible for the transcription is the RNA polymerase. Eukaryotes have three types of RNA polymerase: RNA Pol 1, RNA Pol 2, and RNA Pol 3. Pol 1 for ribosomal RNA, Pol 2 for mRNA, and Pol 3 for tRNA. But then we also need transcription factors, without which the promoters are not identified in the eukaryotes. So transcription factors we will use only for the eukaryotic DNA, eukaryotic transcription, not the prokaryotes. Okay. Then this is a very important thing. We need to know the difference between the coding strand and the excuse me, non-coding strand. Or we need to know the difference between the template strand or the non-template strand, the sense strand or the non-sense strand. So what is a template strand? Template strand is that strand of DNA that forms the mRNA. As you can see here, only this strand is forming the mRNA. This strand is not forming. Okay, so that strand which forms the mRNA is called as the template strand. Okay, and the other strand, right? So the other strand which is not forming the mRNA is called as the coding strand. Okay, and why is it called the coding strand? If you remember, I told you last time that it is identical in the base sequence what we have in the RNA transcript. So this and this, the base sequence will be same. So that is why it's called as a coding strand, even though it does not form any kind of mRNA. Okay, so don't get confused with it, everybody. Remember this. This is a very important point. Okay, so then we discussed that a typical structure of the eukaryotic gene. We said that every eukaryotic gene has two components, a structural gene and a regulatory gene. What is a structural gene? Structural gene is that gene which is transcribed into the RNA. That means this part which forms the mRNA, right? This green part which you see here, okay, is called as a structural gene. Okay, this gene might have an initiation site, as you can see here. From here the transcription would start, right? It might also have a termination site where the transcription will end up, right? And then we have another part in the DNA which is called as the regulatory. So this is the regulatory gene. So what is the regulatory gene? It controls the transcription of the structural gene. Very important point, right? And is this gene transcribed? No, remember this point. This gene is not transcribed into mRNA. It's not there to form the mRNA. Could we say that coding strand is exons? No, 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 no. No, there. Coding strand is just the strand which is a non-template strand, which is complementary to the template strand. No, no, no. Exons, you can say, for example, no, it's not about that. It's, see, basically exons and introns are basically, I can have exons here also. I can have exons and then introns, exons and then introns. And in this one also I can have exons and introns. No ways. So this is basically, the definition is in terms of which strand is forming the mRNA. That's all you have to remember. Which strand is forming the mRNA. Okay, so the strand which forms the mRNA is called as a template strand. Okay, and the complement to it is the coding strand. Is it clear what I'm trying to say? So that definition is based on the mRNA. Absolutely, yes. Okay. Okay, so we were talking about the structural gene and the regulatory gene. So this is a regulatory gene. Its function is to control this one, to control the transcription of the structural gene, right? And this regulatory gene over here, if you see here, it has these Tata boxes, cat boxes, right? It has promoters present in it. So you need to know the definitions. What is a promoter? Promoter is that sequence on the DNA which actually helps the RNA polymerase to bind to it, okay, and start the transcription. So you can say that RNA polymerase binds to this promoter. For example, if this is the RNA polymerase, with the red one, so it binds to this promoter called as a Tata box, for example, right? So when the RNA polymerase will bind to the Tata box or bind to the promoter, then only the transcription will be started. Is it clear? Did you all understand the definition of promoters? Very, very important point, right? Yes, as the most important, most common identified promoter in the eukaryotes is the Tata. So why it's called a Tata box? Because you have thymine, adenine, thymine, adenine, thymine, adenine, which is a repetitive sequence over here. Okay, then we know that we also need transcriptional factors. So what are transcriptional factors? Transcription factors are those proteins, right, which bind to the DNA. Okay, for example, which bind to the DNA, but where? They bind to the different parts of the promoter. For example, if this is my DNA, okay, let's say this is my Tata box or the promoter, right? So within this promoter, I might have certain sequences. We call them DNA binding protein, DNA binding sites. There might be certain sites to those places, to these places. For example, maybe this one in the promoter region, this one in the promoter region. This is the place where your transcription factors will bind. There are many, many transcription factors. We usually write them as a TF, TF1, TF2, TF2A, TF3A, right? So transcription factors, remember that they're also called as the DNA binding proteins. Okay. Now you might have asked me a question that why do we need transcription factors to bind to the promoter, right? I just told you it's the RNA polymerase which binds to the promoter, right? But you have to ask these questions, but you did not, nobody asked me this question, right? So the reason is why we have transcription factors, because the RNA polymerase have very, very less affinity towards the promoter. You can say it becomes blind. It cannot identify the starter box, its promoter. So it takes the helps of what? All these transcription factors. So they bind to the promoters. Okay. So once the transcription factors will bind, then these transcription factors, they attract the RNA polymerase, and then the RNA polymerase can bind to it. Is it clear? Did you all understand why do we need transcription factors? Right? So in short, your promoters are identified by the transcription factors, and they are present only in the eukaryotic genes, not in the prokaryotes. Okay. And then post-transcriptional modification, what is that? Once the DNA is being formed, once the mRNA is being formed, okay, this mRNA needs a lot of things to be done so that you can convert it into the mature mRNA, right, and then export it to the cytoplasm. That's called as a post-transcriptional modification, right? Because the RNA which you formed here is just a non-functional RNA. Okay, it's not it's called the pre-mRNA. Okay, to make this pre-mRNA into functional mRNA, okay, we are doing two things. Number one, we're removing first of all the introns. As you can see here, we'll remove the introns. Okay, and then what happens? As the introns are removed, then the exons are joined back again. Point number one. Okay, point number two, we are doing two other things called as the capping and the polyadenylation. What is capping? Capping is done at the 5' end. This is the 5' end, this is a 3' end. So 5' end, we simply keep on adding, the methylated form of the guanosine. Okay, so we might add methylated form of guanosine from the 5' end, and to the 3' end, we keep on adding adenine. This is just a non-coding sequence. Okay, as you add tail to the mRNA, to the pre-mRNA, and as you cap it at the 5' end, this is basically very, very important, because this will protect the two ends of the mRNA. So mRNA is now safe. It cannot be degraded now, right? It's safe now. Yes. And number two, it will also facilitate the transport of the mRNA to the cytoplasm. Right? So the point number one is, why do we do this capping and adding the tail? As I said, that the most important thing is we need to protect this pre-mRNA, because there are plenty of ribonuclease enzymes which might attack this mRNA, and khalas, it will be lost there, right? So capping and adding the tail basically protects this mRNA. Okay, and then later on we'll talk about how this capping helps in the ribosome binding, also in the translation. That is another use of this capping and the tailing. Is it clear? But the basic aim is to protect the degradations. But you should know all the reasons. Sometimes there's a question, right, which is basically, which would ask you, what are the different reasons? Why are we doing post-transcriptional modifications? So you should know all the reasons. So what are the various reasons? Number one, protecting from the degradation. We don't want it to get degraded. Number one, helps in the export of the mRNA. Number two, helps in the export of the mRNA to the cytoplasm, right? And number 3, what was the number 3? It also helps in the binding to the ribosomes, which we will see in the translation. Is it clear? Did you all understand the reasons behind it? Any question? Yes, everybody, are we all awake? Girls and boys? Is the revision class boring? Because you already took the concept? Okay. Okay, no issues. All clear. Okay, wonderful. Right? So that was about the transcription. Okay. So transcription takes place in, you also need to remember the points of the transcription. As I told you, transcription takes place in the 5 dash, the way we remember the rules of the replication. You also need to remember the steps or the rules of the transcription. Number 1, it takes place in the 5 dash, 3 dash direction, the same as your replication, right? And number 2 point, we have the enzyme called as an RNA polymerase enzyme here, not the DNA polymerase. Then we said that RNA polymerase has no proofreading activity. Okay. Number 4, we said that transcription takes place in the nucleus. In the nucleus. Okay, is it clear? Now we move on to the translation. So what is the translation? Translation is where we form what? Where we are forming the proteins, okay, from the mRNA. That's the translation. Okay. So translation takes place on the ribosome. The site for translation is the ribosome in the cytoplasm, right? So then this is the role of various mRNA, RNA, and TNA. I've already explained you previously. This is just a revision class, right? So we already know about these mRNA, RNA, and tRNAs. What is their role in the translation, right? So yeah, the most important point which you need to remember here is that we have 20 different enzymes, okay? The enzymes called as amino acyl tRNA synthetase, which helps in binding the amino acid or attaching the amino acid to their corresponding tRNAs, right? So if this amino acid binds to the tRNA, okay, this is the tRNA, this is the amino acid attachment side, right? This reaction is catalyzed by the enzyme called as amino acyl tRNA synthetase. So we have specific different types of tRNA synthetases, as we have different types of tRNAs for 20 different types of the amino acids. An important point to remember over here. Is it clear to everyone? Okay. Then the genetic code. Yes. For genetic code, if you remember, I told you that we need to memorize these points. Okay. First of all, we need to memorize how many codons are there. Can anyone tell me how many codons do we have all together? How many codons do we have? 64, right? Out of 64, how many we have the stop signals? Can you name the stop signals? You should be knowing about it. UA, UAG, and UG. Absolutely. Very good, everyone. Absolutely. You're right. Right. And how many initiation codons do we have? We have only one, AUG. Very good. Right. To start the initiation of the translation. Very good. Yes. AUG codes for methionine in eukaryotes. But what about the prokaryotes? Mohammed, it codes for? Anybody? What about the prokaryotes? Because in your book, they have mentioned about the prokaryotes. Formal methionine, absolutely, Shata, very good, right? How many tRNAs do we have, approximately? tRNAs for 20 different amino acids. How many tRNAs we have? No ways. No, 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 no. We have more than 20 tRNAs, if you remember that. For one amino acid, we can have many tRNAs. This is an important point to remember. No, we just don't have only 20 tRNAs. We might have 45 to 50 tRNAs, right? How many amino acyl tRNA synthetases? How many enzymes do we have? And how many amino acids do we have? How many enzymes, amino acyl tRNA synthetases? And how many amino acids, guys and girls? 20. So 20 amino acids and 20 amino acid tRNA synthetases. Very good. Absolutely very good. Right? So what is a genetic code? So genetic code is you don't have to memorize this. I told you last time also, right? You don't have to memorize this. You need to know what is a genetic code. The triplet of base sequence, okay, on the mRNA, okay, is called as the codon, and each codon codes for a specific amino acid. As you can see here, AUG codes for methionine, right? CCU codes for the proline, UAC for the tyrosine, right? So now what you need to know, you need to know about the features of their genetic code. That's very important. We know that genetic code is basically the codons on the mRNA, as I said right now, right? And we need to know it's always 3 bases, it cannot be 4 bases. So it's a triplet, right? So then it's a triplet, right? And then it's non-overlapping. What do you mean by non-overlapping? Okay. So for example, as I have AUG, okay, then I have GGC, then I have CAA, okay. So the codons are read one after the other, like AUG, as I said, GGC, CAA, right? There's no comma, okay. It's unpunctuated. There's no comma. There's no full stop. There's no semicolon present here. So if I have this base sequence, I know that there are 3 codons present over here, 1, 2, and 3, right? So this is called as the non-overlapping. Degenerate. Does anyone know what is degenerate? One amino acid has more than 1 codons, isn't it? If you remember, I told you that people do get confused with the word degenerate. One amino acid can have more than one. Very good, Shata. Very good. Absolutely correct. One amino acid can have more than 1 codon, right? Then you can know the examples, for example, from this. Okay, serine has, for example, 6 codons. You can know all these examples. Okay, serine, arginine, right? So they have more than 1 codons. Okay, or there are certain amino acids like methionine, right? Methionine, as you saw here, it has only 1 codon, AUG. Okay, or even tryptophan. Okay, it has only 1 codon, right? So all these things you can memorize this slide as well. Okay, and then we said that okay, we said that about the degenerate. Okay, and then we also say that it's a universal. What is universal? Universal means that every organism, the monkey and the humans, everybody will have the same genetic code. Okay, so the codons are same for the same amino acids in all the species, whether it's a human or whether it's the animal. Okay, and one more point, it's the genetic code is unambiguous. What is unambiguous? It's not ambiguous, it's not confusing. One codon codes for one amino acid, right? So the same codon cannot code for many other amino acid. So there's no confusion here in the genetic code. And the last is that the genetic code contains start codons and the stop codons, which I already explained it to you. UA, UAG, and UGA, and the AUG is the start codon. So all this you can memorize. Then the process of the translation, the most important, how exactly does the translation take place? We have 3 important steps, okay? One is called as 4 important steps. One is called as an activation, initiation, elongation, termination. Actually, there are 3 steps: initiation, elongation, and termination. But before that, we need to charge the tRNA, right? Or we need to activate the amino acid. So that is why they add on to the, but basically we have 3 processes, these 3 processes. Okay. The first process is charging of the amino acid, or activation of the activation of the amino acid, wherein we have an amino acid, it binds to the ATP and it is activated first. Okay, as you can see here, the activated amino acid. Okay, so once we have the activated amino acid, then what happens? The next step is basically the charging of the tRNA. So what do you mean by charging of the tRNA? If you see here, I told you this is the acceptor arm. This is the, you should know about this. Where exactly does the amino acid bind to? Does it bind to 5' end or 3' end? Should be knowing about it. It binds to the 3' end of the tRNA, which is called as an acceptor arm. Right? Yes, Noor. Very good, Noor. Very good. Right? So if you see here, this is the amino acid, the activated amino acid, which binds to the 3' end of the tRNA. Okay, so here actually it has 3 unpaired electrons, 3 unpaired bases over here, so that's why it binds here. Okay, so because it provides an OH group to it, a free OH group to it. Okay, so that free OH group acts as a binding site where the amino acid can bind. Okay, so that's called the charging of the tRNA, very important step over here, right? And then we know that the ribosomes, so this is a very, amino acid activation is a very important step. Then we know that for the prokaryotes that we have 70S ribosome, for eukaryotes we have 80S ribosome. And if you remember, I told you here it has explained the process of initiation, elongation, and termination in the prokaryotic ribosome. So that is why you will find here a 50S and the 30S ribosomes, right? The smaller unit is the 30S ribosome, and the bigger unit is the 50S ribosome in the prokaryotes, as already discussed. Okay, and then you also know that whenever the translation has to occur, okay, so it occurs on the bigger subunit, right? But then you require both the subunits, but at the end of the translation, the 2 subunits are separated from each other. 50S will be separate and 30S will be separate. Okay, so but once we start with the initiation of the translation, right, you will see that then the bigger and the smaller and the bigger subunit, they will come close to each other. We'll see that in a while. Okay, so they are associated with each other and then dissociate once again at the end. Okay, so now what is the first step of the translation, or you can say after activation or charging of the tRNA, the first step is the chain initiation, or you can say initiation of the translation. Okay, so what happens in the initiation? Okay, as you can see here, the smaller subunit, the 30th subunit, okay, binds to the mRNA. Look at this is the mRNA over here. Okay, and then we also need some initiation factors, IF1, 2, 3, for example. Okay, so you will see that this mRNA will slide through this smaller subunit like this, okay, until the first codon called as an AUG is exposed. Okay, but then if you remember, I told you about the Shine-Delgado sequence. You should be knowing about this. Is it the mRNA? I'm sorry, Mohammed. It's the mRNA. I didn't understand your question. It's the mRNA. Of course, the mRNA has the information, Mohammed, right? Not the tRNA. tRNA will come later on. tRNA will just bring the amino acid. But the mRNA has the recipe to make the protein, isn't it? So it's the mRNA which will slide. Okay? No, don't worry. It's okay. Right? So as I said that this translation is also very confusing among the students. I'm telling you all this based on, once I graded the papers last year, right? So that's why I wanted to revise these 2 chapters, because I know that students do get confused with all this. So What is a Shine-Delgarno sequence? Shine-Delgarno sequence is basically, if you remember that, it is present in the prokaryotes, not in the eukaryotes. Okay. Now, what is the function of the Shine-Delgarno sequence? As you can see here, the sequence is present on the mRNA. This is the mRNA, so that you don't forget it. This is the mRNA. Okay. So SD, or a Shine-Delgarno sequence, is present on the mRNA of the bacteria, of the prokaryotes. Okay. And this sequence is very important, right? Because it helps to in attracting, or you can say in recruiting, the smaller subunit of the ribosome to the mRNA, right? Because I just now told you that the mRNA should slip through the smaller subunit. But this slipping through will happen only if they attract each other, right? So Shine-Delgarno sequence, right? It makes sure that the alignment of the ribosome and the start codon, right? It takes place. Is it clear? So it's basically, again, it might be a sequence. It's not something different. It might be adenine, guanine, guanine, uracil, uracil. It's a sequence on the mRNA, okay, which is complementary, to a sequence present on the ribosome. Is it clear to everyone what I just said? Right? So we definitely, for the initiation to occur, we definitely need the Shine-Delgarno sequence, right? Without this, the mRNA cannot bind to the smaller subunit, which is an important step in the initiation of the translation. Okay? Is it clear? Okay, so where were we? So now we have the smaller subunit, we have the mRNA, khalas, they all are together now. Okay, now what's going to happen? Now we will have the tRNA coming up. Okay, hello, I'm coming. I'm also here. Okay, I brought the amino acid, right? And it is the first charged tRNA, which is the tRNA methionine. Okay, if you remember, I told you it's a tRNA methionine in the eukaryotes, okay, but and that's how we write it, tRNA formyl methionine in the prokaryotes is the first charged tRNA which will come here, okay, which bears the first amino acid, which is the methionine in the eukaryotes and formyl methionine in the prokaryotes. Okay, so what do we have here? Okay, we have now smaller subunit, we have the mRNA, we also have the tRNA, right, with the charged amino acid. And what is lagging here? We have this is the initiation complex. What will complete the initiation complex? What completes the initiation complex? What is the final step of the initiation? What should come now? The larger subunit, absolutely. So the final step in the initiation of the translation is the addition of what? The larger ribosomal subunit. Okay, but at the same time, it will remove these initiation factors which were attached here, So all these factors will be removed once the larger subunit will bind here. Okay, and then the larger subunit, we said that it has 3 sites. Okay, it has 3 sites. This one, the A site, the P site, and the E site. The E site is an exit site, P site is a peptidyl site, and A site is an acceptor site or the amino acyl site. Excuse me. Yes, Mohammed. So the capping send like a message. This is the end of the mRNA in this one. See, capping, as I said, Mohammed, that as I told you initially when we started this initiation, I told you that here we will talk about the initiation in the prokaryotes, not in the eukaryotes. Capping would help the eukaryotic mRNA, not the prokaryotic mRNA. Do you understand what I'm trying to say? So the capping is basically the ribosome will bind to the 5' end, okay, of the mRNA. Okay, so capping would help. Yes, you're right. So capping would help it to identify that, okay, I have to scan from here, So your ribosome will start scanning from 5' end once it knows that, oh, it's the capped end, right? And it will scan, 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 scan till it finds the first initiation codon, which is the AUG. Okay. So the capping is like more importantly in the eukaryotic DNA, right? Here we have mostly the Schindel-Gardner sequence, which is doing the job. Even though we have one more sequence in the eukaryote, we call that Kozak sequence, but you don't need to know all that because eukaryotic initiation is not there in the textbook. You just need to know the prokaryotic initiation, not the eukaryotic initiation. Okay. So then we discussed that what are the three, sites in the larger subunit of the DNA. We have the exit site, we have the peptidyl site, or the P site, we have the A site or the amino acyl site. Okay, and you know that the first charged tRNA will always sit where? In this peptidyl site. The rest, all of them will come into the A site, right? So remember that this is an important point to note down here, right? So the only charged tRNA that enters the ribosome, okay, through the P site is what? Is which tRNA? Is a tRNA F-methionine, okay? tRNA F-methionine. That's the only one which enters here, F-methionine or methionine in the eukaryotes, okay? Then the second one will come into the A site. Okay, so this is already mentioned over here. Now what will happen in the chain elongation? Elongation is simply like, once you have the first tRNA in the P site, okay, now the newly charged tRNAs will come, right? Newly charged tRNAs will come based on what codon we have on the mRNA, based on this codon, what we have on the mRNA, right? Along with the various elongation factors, which you can see over here. They're all elongation factors. And that the newly charged tRNA will enter into the A site. Okay. Then the second step in the elongation is the peptide bond formation. So between the two amino acids, the peptide bond is formed. Okay, between the two amino acids, one present in the A site, okay, the new one, and the other present in the P site, the older one, right? So the enzyme here is the peptidyl transferase. You need to know the enzymes as well. Peptidyl transferase is the enzyme, transferase. Okay. And the third step about the elongation is the translocation. What is translocation? Ribosome will move in the 3' direction. Right? The ribosome will move in the 3' direction. Okay. And here for this translocation, we have an enzyme called the Translocation enzyme. Okay. So as it will move down, so accordingly, what will happen? The uncharged tRNA will move to the exit side, right? And this one will move to the P side. Okay. So one will be in the E side and now another one will be in the P side. And now this place is again empty. Now the third tRNA will come into this acceptor site once again, and the process is going to repeat and repeat and repeat and repeat, right? Unless and until we have a stop codon UAA, UGA, UGA, and UAG, right? So once you have, this is a bond formation over here. Once you have the UAA or UGA, right, or UAA there, UA or UG or UAG there, right? So there is no, remember that, okay? There is no tRNA for that. Okay, rather we have a release factor for that. So there's no tRNA for that. There's a release factor which will come in this A site, which will give the information to this ribosome that, okay, we are done. We need to cleave, we need to break, or we need to remove this amino acid chain. Okay, and then once this is released, then it will be folded into, or tertiary structure, quaternary structure, whatever structure it has to be, and that and the ribosomes will again dissociate. Everything will dissociate. Look at this. I have the mRNA, right? I have what, the tRNA, and then we have the two ribosomes will separate from each other. Is it clear? That is that's how the termination of the protein synthesis will take place. Okay. So gene regulation, if you remember, I told you that for the gene regulation, it can take place at the transcription level as well as the translational level. Transcriptional level, we have these, these transcriptional factors, right? We have the enhancers, we have the activators. We spoke about them previously, right? So that was about that was about how the gene regulation would take place. Okay. And then how do they bind, how do transcription factors bind to the DNA? They have special kind of the motifs, okay, or you can say they have special kind of places which can attach themselves to the DNA, right? We spoke about the metal binding fingers, also called as the zinc fingers. If you remember, we spoke about it, right? So how they, and as you can see over here, how these zinc fingers, okay, they interact with the specific site on the DNA. Okay, they recognize, okay, the specific hydrogen bonding on the DNA, right, and bind to it. Okay, so we have, these are called the zinc fingers or metal binding proteins, right? So this is their specific sequence of the amino acids. They have zinc in the middle, right, and all these amino acids this way, right? So that's how your transcription factors bind to the DNA. That's why they are called the DNA binding proteins. Okay. And then alternate splicing. This was an easier one. We simply said that how, there are different ways how mRNA transcript can be spliced, okay, so that, you can produce the different proteins from the same genes. That's called an alternate splicing. You can splice it this part, okay, you can splice it only this one and this one, okay. So there are different ways, by which eukaryotic RNA can be spliced, right? And this is called an alternative splicing. So by the process of alternative splicing, an organism can produce more than one type of polypeptide chain. As you can see here, see how many types of mRNAs are produced from the same transcript by doing the alternate splicing. Is it clear to everyone? Everybody? Any questions till now? And this is the same thing, base excision repair and the nucleotide excision repair, which I explained it in the previous class, right? In the lecture, in the morning lecture, right? In the morning revision lecture, this is the same thing. Okay. Then what is the recombinant DNA technology? Okay. If you remember, I told you recombinant DNA technology, wherein we can produce an artificial DNA, okay, by taking the DNA from different sources, okay, and when we combine those DNAs, it gives rise to one single DNA, okay, which is called as a recombinant DNA. Okay, so you can create it by doing what? By taking the DNA from different sources and combining it, right? And then this DNA is incorporated in the plasmid, right? Then if you remember, I told you that this cannot happen without a specific scissors, the way we have scissors to cut something, right? Especially we have the restriction endonuclease enzymes, okay, which help in cutting the DNA, right? We call them restriction endonuclease. We call them molecular scissors. They are there to cut the DNA, okay, and exactly at the same position, but the cut is staggered, as you can see here. It cuts like this. Sometimes if you have a carrot and if you want to cut the carrot, you put a staggered cut, not a straight cut like this. So restriction endonucleases are specialized in putting the staggered cut, which can produce the sticky ends that can be easily used to incorporate the DNA into the plasmid. right? If you, I gave you an example about how humulin was produced, how the artificial insulin was produced using the recombinant DNA technology, and then I explained you that whatever vaccines which we have used nowadays to treat the coronavirus, they also have been produced using the recombinant DNA technology, wherein we definitely need a recombinant DNA, wherein we need a vector DNA like a plasmid, right, in which the DNA can be incorporated, right? And then we need, as I told you, we need the restriction endonucleases, which can cut the DNA. Then we need the DNA ligases, which can stick the DNA. And then this plasmid, once this plasmid has our gene of interest, right, it can be incorporated back into the bacteria, and bacteria can produce the clone of it. Okay. Then the gene therapy, we spoke about what is, you need to know what is the difference between the ex vivo gene therapy and the in vivo gene therapy, right? So in vivo gene therapy, wherein we directly deliver the genes into a particular tissue. Okay, so viruses are used to infect the patient's tissue. That is in vivo, within the body of the humans, as you can see here. And then what's an ex vivo? Ex vivo is basically you take the genes from a particular person, right? And they are cultured in the lab. Okay, and once once we have those cultured cells in the lab, right, then you incorporate the missing gene. The idea is to give a correct form of the gene to a particular patient. We call it a gene therapy, right? And how this missing gene is incorporated by using we use a virus, okay, to incorporate that gene into these cells, and these cells can be transplanted back into the human being. So we call it an ex vivo gene therapy. Okay, we have incorporated gene outside the patient's body, right? So that was all for this chapter. This was the last slide for this chapter. So any questions from anybody? Do we have any questions about it? Yes, boys and girls.